Welcome everyone to the first DataBeers Brussels.
Yay!
Okay, hello everyone, and welcome to the 16th edition of DataBeers. I will give me just a second to share my screen. I can't seem to get used to this. Voila. So it's the 16th edition. Can everyone see my slides?
Yes.
Invite the panelists to tell me. So it's a bit odd online. We can't wait to see you all again at Muntpunt. But meanwhile, we'll have to do it online. So what is DataBeers? DataBeers is an initiative that started in Madrid quite a while ago and then spread all over the world. The concept is 4 or 5 short talks, 7 minutes about data, no formulas, no advertisement, informal setting. You get free beers in a live setting during the event, and afterwards we typically go down for even more beers. But so for now, how it's gonna work this time? So through Zoom, you found us, of course. So grab yourself a beer like we did. If you have questions, you can use the chat and maybe raise your hand if you want to talk. And also we'll ask some questions before each talk in a poll, and we'll do the same right now. So to know a bit your background, could you present yourself? So what is your background? What do you work in industry? Whether you work with data at all, or you just need an excuse to open a beer. Voila. I'll wait a while. It's much easier to control the crowd now. It's very quiet, very good, because the acoustics in my living room are not that good. So, well, that was the first bad joke of the day, probably, yes. Okay, so we'll discuss the results. Let's have a look at. Yeah, voila. Thank you, Charlotte. So we see that most people here are working in academia and with data. Very few people crashing the party, only 2 out of 30, not bad. And so pretty much everyone works with data. Nice to see. Then we'll also use the chat. So if you could now enter in the chat what beer you're drinking, where you live, and actually, I know everybody thinks Belgium is an ugly shithole, but look at this. This is selected the most beautiful city in Flanders, Reken. So if you don't know where to go, that's a place to go. And then
it's
also
hellhole, Johan. It's hellhole. That's what Trump said.
Hellhole. Yes. Sorry. It's a hellhole. That's why you see this water on the picture. And then the third element in the tuple. Can you guess the name of this dog? So if you could enter the name of the beer you're drinking, where you're living, or where you currently are, and then the name of this dog. So the first name is fair enough. Okay. Then more about us. So we're a team of about, I forgot Jose, shit, sorry. So we were a team of 7 main organizers, and we're recently joined by Jose, who will introduce one of the speakers. So it's a good opportunity to get to know him. And who are we introducing or seeing? Who's talking tonight? So we have 4 speakers. We'll start with Juami from Vrije Universiteit Amsterdam. Then we'll move to Joris, then Milena, and finally Sam. So it's a tradition that we tell a little data story. And so I took the time to look up in a very reliable source what data is. So I went to the Urban Dictionary. And so I discovered interesting things that I didn't know about data. So data is, of course, the plural of datum, which means a guy who's considered a stud. Interesting. Or someone who gets an erection from listening to classical music. This gets more personal then, of course. Data is, of course, a character in Star Trek as well. And I doubt that this is really a quote from Data. And it's also pictures that girls take of themselves being naked, really. Interesting. I also learned that there's more metadata than data. Interesting. And that metadata have nicer pictures if you do an image search. Also, I learned about circles in metadata that completely make no sense, and a sad metadata kitty, which is also completely new. That is often very sad because there are duplicate records, because she had to add a death date to an NAR, or because she built a house that no one sees. So if you're working with data, I beg you to sometimes think of sad metadata kitty. So I hope you have a better story. And if you have a better story, don't hesitate to contact us personally or through the site or on this email address by sending an email to this address. So I also want to thank our sponsor, Business and Decision, Muntpunt, where typically the live events are taking place. And then please do not forget to fill in the survey that we'll send tomorrow so we can optimize our events. And also we will send beers to the top 5 people with the best questions. We'll send them only in Belgium, and or we can deliver or have a drink with you when it will be possible again live in Brussels. Okay, that's about it. I'll leave the floor to Milena, right?
Hello, everybody. So welcome to the 16th event, Second Times Online. So our first speaker today is Juami. And Juami is recommended to us by our new team member, Jose, who pitched her as the smartest person he knows. So I'm going to let him present her now. Go ahead, Jose.
Well, as Yelena has already said, there's something in my face. Oh, this. I decided I suggested Juami because she's a very dear person to me. I met her when I was living in Amsterdam. We were office mates at the Vrije Universiteit Amsterdam. And I met her when she was doing 2 masters, one in biomolecular sciences and another one in bioinformatics and systems biology, for which she graduated cum laude for both of them. I think that already goes in line with what Yelena said. I think she's probably the smartest person I know, and she's one of these people that when she lands in your group, you just cannot let her go. And that's exactly what they did in Amsterdam. I mean, she stayed there. She does everything. Intensely everything from research to dancing to watching multiple seasons of a show in one day. And well, currently she's doing her PhD at the Vrije Universiteit Amsterdam in the bioinformatics department. And please let me introduce Juami van Gils, and she's going to talk about her work with neurodegenerative diseases and the struggles that she finds with bioinformatics, that is, and how she deals with them.
All right, thank you, Jose, very much. So I can now see the poll results. I'm not sure how to do this. Let's see. So about half of you have had a relative or know someone who has dementia, which I think kind of indicates how prominent this disease is. And on the second question, I want to come back a little later. I'm going to leave it open. Can we leave the poll open for a second? Oh, no.
I can share the results later again when you need to have them.
Okay, perfect. All right, let me share my screen. All right, so you should be able to see the presentation now.
Yep, perfect.
Yes, all right. So Jose, thank you for this introduction. And so what I will be talking about to you today is a project that we got from the medical center here in Amsterdam. And we got a question from this medical group, experimental group that had a data set. And they could do some basic analysis with it, but they're not computer scientists. So they asked if we could do maybe some more advanced analysis on this project. And this project has to do with frontotemporal dementia. So I think the first question, yes. So do you have a relative or know someone who has dementia? That was about half of you. So diseases like dementia, but also diseases such as cancer that are very common. The kind of data sets that we get to answer questions about these diseases are quite similar. And there's some very common struggles that we as in the field of bioinformatics run into when we try to answer these questions. And so what I would like to do today is use this project that we have for frontotemporal dementia as an example of these struggles, because I think like they're very common in bioinformatics, but in computer science, they're maybe not so well known. So to do that, I would like to take you back in time first. So in 2001, the first complete human genome sequence, first complete human genome was sequenced. So this is about 3 billion base pairs, which contains the genetic information of a person. So this is unique to every single person. And now in 2001, we thought, okay, we have this data now, we have this information. Now we will fully understand human biology, how everything works. We'll be able to solve every disease with this knowledge. And then maybe that's not exactly what happened. So we're now 20 years further and we still have these kind of diseases. So what happened? And so why is this problem actually so much more difficult than they thought this would be? So first I would like to give you a bit of a feeling of what kind of data we have. So the general process that we're looking at, we have this DNA sequence. The DNA the genes on this DNA, and we have about 20,000 of them, are transcribed into RNA, which is can, in the most simplest case, be seen as an intermediate step to make proteins. And these proteins are the actual functional molecules in your cell. So these proteins, they make sure your muscles contract, that you can digest food, that you breathe, that you can think, so the neurons in your body, like proteins are involved in all of these processes. And so if you have, for example, DNA, the kind of data that you get out of is this DNA sequence of either a gene or maybe the complete genome. What you can, for example, say is if you have the genome of a healthy person And if someone is sick, then maybe they have a mutation somewhere that's causing this disease. And so we can, for example, see where these mutations lie and then try to determine if maybe or maybe they do not have an effect on this disease. For RNA and also for proteins, we get expression levels. So what we do is for the different samples, so these are typically people, patients, for example. And for every patient, for a number of genes, we determine the level of either the RNA or the protein molecules in their cell. So we have these 20,000 different proteins or genes. And every, in a typical cell, every one of these proteins has a value that's either 0, so then it's not active at all, or maybe a low level or a high level. And this is, the higher the level, the more active we say that this gene or this protein is. And so we can measure these levels in a tissue type or a cell or a person. And then we see, then we try to say something, usually, for example, try to compare two groups or a healthy group versus a disease group and see where are the differences. And so indeed, so these protein levels are usually used as a measure of activity. And so for this project in the FTD case, we're looking at protein levels, just for your information. Then I would like to get back to the second poll question. So what do you think the typical size of biological data set is? So some of you, so the largest answer are about 10,000 samples, 10 to 20 features. And I also see 30 samples with, so basically the opposite, 30 samples with 1,000 to 20,000 features. So the one on top, those are probably the people in bioinformatics, because this is the actual truth. So we usually have about 30 samples, with then in the case of proteins, usually about 1,000 to 3,000 features. If we do the RNA, that's a little bit easier. So we can measure more, you can end up with 20,000 features. And now I cannot see a lot of you, but for some of you, that looks a little bit like this. And so what I want to do in the rest of this talk is to explain to you why we have this problem in the first place, and to try to convince you that we can still actually learn something useful from this data. So in my case, going back to the frontotemporal dementia, our data set had 26 samples, 5 from control, 6 from one frontotemporal dementia subtype, and 15 from another subtype. We had about 1,800 proteins that were measured. And so these samples, they actually come from the human brain. So you take a small section of this region here in yellow, and then we measure the proteins in the brain section, and then we do the analysis on that. And that's exactly where the problem lies. You need human samples for this data. So in this case, it's very invasive to get the sample. For this, even the patients already need to be deceased and have donated their body to science. You cannot do this on a living person. And so it's very difficult to get these. There's also a lot of ethics committees that make sure that you really need this data because it's very rare and very valuable to get. But even if you say you can do this on living people, for example, a sample of cerebral spinal fluid or bone marrow, it's very painful and very uncomfortable for patients to donate. So you just don't have a lot of this data. And even in the case if it's more simple, so for example, if you take someone's blood sample, that's relatively easy, you still have privacy issues. So for example, if you sequence someone's genome, that's very personal. And for example, insurance companies are very interested in this information. So you have to protect it very well. So for example, hospitals, they do a lot of this research, but then the data is kept within the hospital and other people cannot access it. So even if this data is there, you often, it's still very difficult to get to this data. And so we often, if you can get, let's say 50 samples, you should be very happy in biological data sets. And so we have this, now we have this problem. What do we do with it? So what we do to still get some meaningful results out of this data is to use pre-existing knowledge about relationships between groups of proteins. So one way to do that is to take gene sets. So for example, if you have a bunch of genes that you know are related to, for example, metabolism, so the processing of food by your body, what we do is for all the proteins that we measure, you would give them a score. For example, we measure their level in our dementia patients, and we measure the level in our healthy patients. You divide it 2 and you take the log. You get a score that basically says if it's positive, then it's higher in dementia than in the healthy ones. If it's 0, it's about equal. And if it's lower in the dementia case, then you get a negative value. You rank all the proteins high to low, and then you basically just tick them off. So every time a gene belongs to metabolism, is involved in metabolism, you tick it off here in this second row. And what you would do, finally, to see, let's say, if there is some kind of enrichment for example, more activity of metabolism in dementia than in healthy patients, every time you start at 0, every time you find one of these genes, you add a little bit of a score. And if a gene is not involved in this gene set, you subtract a little bit. And so if there's a lot of genes, so for example, here at the start, you see there's a lot of genes that are involved in metabolism, the score goes up a lot, and then gradually it goes down. And what you would then in the end use as a score is this final difference between the final maximum deviation from 0. And so if nothing's, if it's about spread equal, and so nothing, this gene set's not particularly enriched, it would sort of stay close to 0 a little bit. So it would go up a little bit, down a bit, and you would get a low score. And so what we can say, if the score is very high, then probably in this gene set something is going on. It's the other way around, if it's very low, so it's lower expressed, you would get something similar. Another thing we can do, so the problem with this is still we have a group of genes. And we know if this group of genes is enriched or not, but we can't really say anything about the relationships between the genes or the proteins themselves. So another thing that we use is a pre-existing protein network. So these can actually be physical interactions, so proteins binding to each other. And it can also be pathways. For example, if you think of metabolism, it's one protein converts a molecule, then the next protein takes the output of that previous step and uses it to convert that molecule into something else again. And what you can then try to determine is if, let's say, a small subnetwork, so here indicated in red, is significantly higher or lower together. And what you can find out by this is, so what you often see in biology is not one protein that has a major effect on a certain disease, but maybe it's a couple of proteins that have a small effect, but that together really have this impact on human health. And with these methods, you can identify processes like this. But so what you put in is the interactions that were already determined by experiments before. And so because we already have this knowledge, we can actually, even though we still have a small amount of samples, get something useful out of this data set. And so, for example, in the case of dementia, what we found is vesicle formation and synaptic signaling. And what that means is that, so we see here 2 neurons, and if you zoom in on this interaction, so between the 2 neurons, then there is these small vesicles. So these are basically little boxes that contain signaling molecules that can be taken up by the next neuron. And so your neurons use this to transmit one signal very fast from one neuron to the next one. And so these molecules are released and then picked up by the next neuron. And what we found is that in one of the FTD subtypes, this process is a lot less active than it would be in a healthy case. So one important thing to notice, and this is my 2 more slides after this, is that these results are often published as such, but there's still a lot of research to be done after this. So we identify basically which pathways or general functional groups are affected, but you still need to do actual research to, for example, more experimental research to really determine what exactly on a molecular level is going on in these diseases. And so why is this useful? And this is one of my favorite papers. It's called Can a Biologist Fix a Radio? And it's a really nice illustration for data scientists to sort of get an idea of how classical molecular biology looks at these problems. So and what happens is a biologist has a radio and it's not producing sound. So the sound is not working. And so how would a biologist go at solving this problem? And that what a biologist would do is get a bunch of radios that do work, open them up, and then one by one, take a component out, see what it does to the radio, and then put it back in, take out a different one. And so what you would do, for example, you take out one component, maybe the volume button doesn't work anymore. So you know that's important for the volume control. You put it back in, you take something else out, maybe the display doesn't work anymore. And based on doing this one by one, a biologist would try to determine what the relationships are between different components. And so a biologist may get something like this. So you know approximately which main components are connected, but you're not entirely sure about the steps in between, how many steps there are. For example, one of these arrows can still be 3 steps instead of just 1. Whereas if an engineer would do this, he would just make a very clear map of how this radio is composed and how it's working together. Now, it's important to note that maybe if a system is small enough, a biologist may still end up with this, but biological systems are often very complex, and so you cannot do it like this anymore. Yeah, so the system in biology is definitely too complex for this. And so one of the things I would like to show you is, and so this approach helps experimentalists to point, okay, maybe we don't know exactly what's going on, but this is like a good starting point for further studies. And so one of the things I would like to show you, this is a human metabolic network. And this, I have to say it's a little bit outdated. It's from about 10 years ago. And so this is 10 years ago what we knew about human interactions in metabolic networks. So it's not yet for, it doesn't involve neural interactions. It's not for muscle contraction. It's really just the processing of food, mostly sugars, in your body. And it looks something like this. Now, every little node here is a molecule, and every arrow indicates the work of at least 1 protein. So it can also be 3, 4, 5 proteins in one of these arrows. And so this network is, I think, about 10 times bigger now, but for obvious reasons, nobody really wants to remake this figure. And so, of course, if you want to take out every single component in this network to figure out what's going on, that's going to take you decades or centuries. Now, the last one, so what we can do is tell biologists, okay, start there. Based on this analysis, start there because that's most likely where something's happening. And look there for probably you'll get some results if you start your research there. So the take-home messages, biological systems, there's no really intelligent design. This developed through evolution over millions of years. And so it's a bit messy. There's no clear, secondly, biological samples are difficult to obtain because these procedures are invasive and there's huge privacy issues in this process. As a result, we have this problem where we have many more features than we have samples, which is not ideal. But because we already have a lot of pre-existing knowledge about relationships between proteins and genes, we can still use this to figure out what's going on in diseases. And so the outcome of these kind of analysis is not a final result, but it's a great starting point for experimentalists to continue the research. And so with that, I would like to thank everybody involved from the university and from the hospital. And I'm happy to answer any of your questions.
Thank you, Juami. Okay, so do we have some questions from the audience? I think I see one, but it's about beers, so that's not really a question. Jacobo, so Charlotte, can you let Jacobo ask his question if he wants to? Okay.
Yep, he's allowed to talk now.
Okay, hi, Jacobo, can you please ask your question?
Yes, so my question is about one problem you mentioned in your presentation is about having a lack of samples, having few samples, but a lot of features, a lot of variables. Are you aware of some technique like data augmentation that can be applied in your field that can solve this problem of the lack of samples, or do you see any solution to this problem?
So what exactly do you mean with data augmentation?
The fact that, for example, you can generate synthetic samples that has properties that are kind of similar to the real sample you have, and that using this synthetic sample can improve the performance of the technique you are using. Usually it happens in the case of machine learning for images, sound recognition, for example, time series analysis as well.
Yeah, so simulated data is used sometimes to initially test models. But one of the main problems is that there's a lot of biological variation and a lot of technical variation in biological data as well. So there's a lot of noise, and so really creating, let's say, augmented data that's reliable is also very difficult. So usually, even if people do that, then in the end, they still, let's say, reviewers for a paper, for example, would still want to see this on an actual data set, even if it's smaller, because that's generally more indicative of how the model is really working.
Okay, thank you.
You're welcome. Milena, you're muted.
Do we have more questions? Okay, I'm. Yeah, maybe somebody of on the panel.
Has a question. Lito has a question. He posted in the chat.
Okay, Lito, go ahead.
Okay, I don't know if I really want to ask this question, but anyway. So okay, in research, we spend a lot of time, trying to find ways to prevent people from dying. And here at the beginning of the talk, you talk about the high prevalence of Alzheimer's disease and dementia. And so I was just wondering, do you think this is a sign that we're just living too long? And at what point do we say, how can we? What's the way that we can die that's okay?
Oh, that's a very philosophical question.
I think Tyrion had a good answer to that.
Right?
Tyrion Lannister had a good answer for that.
Oh, Game of Thrones has been a while ago. Do you remember what the answer was, Milena? What do you say?
I don't think it's appropriate for the wide audience.
Yeah, I think even it's also about quality of life, right? Because, for example, these dementia patients, that can take a long time, and before people actually die, people can have dementia for years. And so, would you still even so, you would still want to if you prevent this disease, people can still get old, and in the end, maybe, in the end, they will die of something. But they will still, for example, be able to recognize their children and things like that. So it's finding cures for these diseases, not just about prolonging life, but also about making life better. So I think it's very important to do this. On the other hand, secondly, I think there's also, for example, early onset variations of these diseases. So actually, frontotemporal dementia is one of the early onset ones that can already affect people in their 40s. And so even if maybe you, in the end, let's say it would happen that we can cure anything and people's life can be extended indefinitely, and we have to decide when we would stop, you would still want to understand these diseases to still cure people at a younger age. So I think this type of research will still be relevant for a very long time before we actually run into those kind of problems. Does that answer your question a little bit?
Yeah, thanks. That's a really nice answer, actually. Thank you.
Okay, so I think this is a good moment to move to the next speaker. So next speaker is Joris. And thank you, Juami. So Joris, okay, while we are getting a poll from Joris, I'm going to present him. So Joris started his career by doing a PhD in machine learning and data science in KU Leuven. However, he was smarter than some of us, and he got away from it on time and moved to industry. And he's been working as data scientist for a couple of companies, Telnet, DataShift, and at the moment he's a freelancer. And his mantra is creating value through data. So not just using this hype to produce any kind of knowledge, which might or might not be useful, but actually create something valuable. And this is something he's going to talk about with the title of the talk, which has all the hyped words we could find, ironically, but let's see what is him. So the poll question, I can also answer. So have your colleagues built algorithm insights, ended up building something under full potential? Why weren't those algorithm insights useful? How much time you spend on those algorithms? How much time you spend? I don't know if it said that. And were those algorithm insights really that great if they ended up not being used? Okay, so can we see the results? Thank you, Charlotte. Joris, I'm leaving you with this and you can start your talk.
Yeah, so to me, the results of these polls are very interesting, especially the last one, because this is kind of a trick question, but we'll come to that. So yeah, lots of buzzwords. Yeah, it happens when you go to industry, I guess. You learn to speak buzzwords. I'm gonna try to just use the useful insights out of these and how they can help us to solve more relevant problems as data scientists or people that work with data science and data in general. Yeah. So my answers to these questions are the following. So yes, I have many examples of cases where I built an algorithm or had some insights that didn't reach its full potential. One that I'll talk about here is one at a telco that I worked with here in Belgium. We had viewing data, and we used it to build an algorithm that reduced our TV advertising cost by 15%, which is a huge deal. We advertise a lot. Why is it why didn't it reach its full potential? Because we only used it internally and never turned it actually into a product, because this is a lot more valuable than just using it for yourself. I spent more than 2 years on this, and not me alone. It was me and many colleagues. So why is this to me a failure? Or why is this not a great algorithm? Because yeah, it probably costed more than it ended up saving. So this was something that I did when I just got out of academia. And this really got me to thinking, okay, why does this happen, right? And to answer this, I'm going to go to a typical data project where some question get asked. It can be from business, from data science, from a research question, whatever. And the data scientist or data person in general starts investigating. So you start selecting data sets. You start taking maybe samples from those data sets because you're working on a sandbox environment. You start building algorithms, discovering the data. And then you get some exciting results. You pour them into a nice looking PowerPoint. You present them to business. They like it, they approve. And then, yeah, the whole thing needs to go to production. So the engineer starts building. When it's finished, it moves to operations. And then the user see it and problems. Can be that maybe the insights were not as understandable as you thought, or that they are not integrated in the right systems, or a bunch of other problems. And what I'm saying here is that not that you can avoid these problems, they will always happen. We cannot predict what our users will want. The problem here is that they end up at the end of the chain. We do all of this work without actually validating our assumptions. And that's what I will take away from the startup. Basically that they say, okay, test your assumptions early. They have this thing called the build, measure, learn cycle, where you select an assumption and hopefully your most difficult, most important one first, build something that allows you to build an experiment, basically, build something that allows you to test it. A lot of the time they call it an MVP, a minimal viable product, which is like a very light version of the thing that you want to achieve, but it still allows you to measure the response. Then you measure, and this is something we as data people should all love, because this is actually, it's a data-driven approach to to innovation or product development or whatever you would call it in general. You take conclusions from these results. Could be that your assumptions were total garbage, could be that they were brilliant, but most of the time it's somewhere in between, that part of it was right, but you also need to adapt. And then you adapt, you repeat, so you keep on iterating this. Back to my story, we actually kind of did this. So again, we were optimizing our TV campaigns based on our viewing data. So one assumption that you make is, can we actually measure the performance of our campaigns using our viewing data? Because in general, there is this body that actually measures all of the ratings, and that's being used right now. Can we use this alternative source of not a panel of 1,500 people, but like a set of 1.4 million people? Is this set valuable enough to actually measure everything? So we had our viewing data, that was great. We didn't have any advertising data. We didn't have any prices for available slots. We didn't have any campaigns. And then you can start shopping basically for data, and you can start bullying people into giving you data. Most of the time that's very time consuming. So what we did was we just faked everything that we didn't have. We were very upfront about it, but this allowed us to build this MVP. So it's allowed us to build a visualization which showed the performance of campaigns. It also allowed us to show these visualizations to the people from marketing and actually get to the right metrics that they used and so on. So this allowed us to validate our assumptions and on the performance measurement. And we did also the same for optimization. So for example, in the optimization problem, it's actually two problems. You need to predict who's going to view what, because you want to predict campaigns in the future. But you also need to optimize. So to test the optimization without the prediction of the views, we just acted as if we had a perfect prediction. So we just used the historical data that we knew basically, and it allowed us to test the optimization and so on. So we did this assumption by assumption, and this was really what allowed us to go to this 15% gains, which is amazing, but still this question why, right? Because I started off the story with that the thing was a failure. Failure is maybe a strong word, but we could have done a lot better, or maybe we shouldn't have started at all, because the assumptions that we were testing were all kind of solution assumptions. How are we going to solve this problem that we have? What we didn't test is our problems. Basically, is optimizing a campaign the right problem to solve? You could think it's a no-brainer, but actually it ended up not being the right problem to solve. And that's what I take away from design thinking, where they, this is the double diamond of design thinking, where they really split the innovation cycle into two phases. First, you have a problem phase, where you focus on solving the right thing. And only then do you have the solution phase, where you focus on solving things right. And it's a double diamond because it has this diverging and converging pattern. First, you use techniques like workshops and so on to select a whole diversity of problems. And then you will devise experiments and collect data to converge to this one specific problem that you think, okay, this is the right problem to solve. Again, this is a data-driven method, which as a data person, I really like. This is really using data to select what problem to solve. And then only then, when you have one specific problem, then you're going to go in the solution. And again, you have this diverging and converging pattern. Here you could say, okay, but I have selected this problem. I know that this is a relevant problem, but it could very well be the case that it's unsolvable. So it's basically bullshit, right? What you've just done. And this is partly true, but the key takeaway here is that spending some time on making sure that your problem is right and then finding out that it's unsolvable is a lot less time consuming than spending 2 years on making the perfect solution for a problem that's just irrelevant. And that's really the thing that we, as mostly technical people have to take away from this is that we are so eager to dive into the technical details that we often forget about taking step 0. Basically is, okay, making sure that is my problem actually, can I prove, not making sure, but can I prove that my problem is worth solving, and can I prove it with data? So basically, back to the story, questions that we only asked after the algorithm was built, but we should have asked before is, okay, who is currently planning all of our campaigns? Do these people, so those are our users, basically, or we should replace them completely, but that's always hard to do. Do they really want an algorithm to do the planning for them? And here the answer was no, because planning these campaigns was their livelihood. They were very skeptic towards algorithms, and because they had such a important step in this entire system, they could resist a lot. And they came with questions like, okay, how can we integrate with all of these systems? How are we going to price these, and so on? And all of these questions were so hard to answer that in the end we ended up not making this into a product. So maybe as a conclusion for me, that like I said before, we are always very eager. So these questions are very hard for us to answer. It's not in our comfort zone, but I think as data-driven people, it's really our responsibility that when we get a problem that needs solving, we first take the effort to try to prove that the problem is actually worth solving. So thank you, and I'm very happy to take questions.
Thank you, Joris. So yeah, what I forgot to mention that Joris is going to become father really soon, like maybe any minute now. So if he disappears, so be very fast with your questions. So let me know if do you have some questions. I'm trying to check the chat. Okay, I see something in Q&A. Vsevolod, I answer live. I think I allowed you to talk. If I didn't, Charlotte, please do it for me.
Yes, I did.
Yeah, it works. Thanks and hi. So basically my question is, don't you think that the adoption of whatever solution you're bringing, whatever solution you are solving, partially comes from the data scientists themselves? So these days, whatever the publication we take or a press release, what we will see is a ton of highlights, how perfect the solution is, how nicely it will perform in reality. But whatever solution will be, there will be definitely some drawbacks, and people are not really brave enough to push it further, and thus potential final clients have some too high expectations, which are rarely met in reality.
Yeah, so there I fully agree, and so there is so much hype around data that basically we are as data scientists, we're getting lazy a bit in terms of, okay, we have this cool thing that we can have a press release or whatever, or a publication, or yeah, you can name it, and everybody will just basically take it because it's cool, it's hype, we're doing cool new stuff, and I think we as professionals, we should be a bit more strict to ourselves and yeah, don't do this basically, and make sure that, yeah, cross all the T's and dot all the I's basically before you yeah, before we do something like that. So I kind of agree with what you're saying.
Yeah, so, and yeah, maybe I would propose that we start to be a bit more educative towards the general public, and when we see that something shouldn't work because of whatever reasons, then we need to try to explain to people why it's like that.
Yeah.
Because otherwise, yeah, you will see a ton of solutions, everybody proposes everything, but in reality, it just can't work.
Yeah, or it works, but it solves yeah, a problem that's maybe not that important.
Yeah.
Okay, thank you, Vsevolod. And any more questions? So 2 new messages. Let me see. So Anton, do you want to ask your questions live? You can unmute yourself. No, okay. So Anton, I'm going to read the question from Anton. Is it actually a task from technical people to ask those questions to themselves, but not for business people?
I think it's a joint responsibility. So we need to be aware that we're in a highly technical field, and it's very hard for everybody to understand everything that we're doing. So to me, it's super easy to go and say a bunch of buzzwords in a business meeting, and everybody will be happy, basically. And yes, this is a problem from business side because they should be more knowledgeable. There I understand, but I think it's also a reality that they cannot be fully knowledgeable about everything. So I think it's very much a shared responsibility, and you see this a lot in very hyped fields. Also, these design thinking approaches and so on, they don't come from data science, they come from the digital sphere, basically, partly, or they are used a lot there. And this is, I think, a field that's like a couple of years ahead on the data field. There was also this buzz in the digital, everybody needed apps, everybody needed websites. And now they are starting to get a lot mature. So I think we can learn a lot from watching those fields, basically, on how we can do better in the data field.
Okay, thank you. Eileen had a question?
Yeah, thanks a lot, Joris, for your talk. I was wondering about this design thinking and the diamonds and diverging, converging. How do you choose what is the right problem to solve?
So basically, for example, you can basically figure out who is your user, who is your beneficiary of your problem, and you fake basically you can go and do surveys. For example, if you have these tools, and I won't do name dropping, I'm not involved in them at all, but you have these really nice surveying tools that you can use to ask questions to the wide public and quickly get answers. So that's one particular way for the problem. So basically, I could say, okay, I'm going to become a dad now. It's a huge problem that X, I don't know what. And because I'm so into kids at the moment, I overestimate the importance of this this problem hugely. What you then can do is just ask people, go and figure out who will be my audience, basically, or my user, and go and do surveys. When you're in the solution space as well, even before you actually develop a solution, you can do mock-ups. You can fake your solution and show it to people, and they will start using it. For example, if you would do a recommender engine, for example, to take a data science example, the assumption can be that, okay, people want recommendations. So then you can first go and ask, okay, do you find your content on a certain channel? When they say no, then you can start proposing solutions for them to actually find it, but actually just build a fake solution. And it really shows you that people will figure out, hey, but I can't use this because, or this is annoying, or you will get some really honest feedback, and it will maybe force you to think in a different way. Okay, how can I use these results from my recommender engine in a good way? And if you cannot find a good way to use the results from your algorithm, then maybe it's not worth building the algorithm at all, because you can build a awesome recommender engine that does perfect. But if you cannot find a way to bring the results to the people, then it's just not worth building.
Okay, thank you, Joris. I think we can wrap up here. So
thank you.
We do the.
Now we can drink beers, right?
Yeah, well, yeah, of course. So I suggest we take a little break. I'm pretty sure after all this beer, we might need like a toilet run or maybe refilling our beers. and we just leave everything like it is. So for the audience, don't go away. We have two awesome talks after the break. It's Milena with her insight from machine learning into depression. And finally, for all of you who can't wait to talk about pandemic going on, we have the really good expert here. So Sam is going to bring some actual true insight in what's going on. So see you guys at, so 5 minutes. What do you think, guys? It's 5 minutes enough?
At 9, we begin again. Sounds good.
Okay, 9 o'clock, we begin again. Okay, so let's slowly come back. So I'll start by reading a bit of the chat. So let's see who we have here. So there are a lot of people on YouTube. I think we did send them a link if they want to join to Zoom. Charlotte, can you confirm that you send them email?
Yes, so I sent an email to all the people who subscribed.
Oh no, I can't hear you.
Yeah, so I just said that I sent an email to all the people who registered to the events, so YouTube and also the webinar, that we had still room and that they could join us online on the webinar if they wanted to.
Okay, so who we have here? We have somebody drinking Victoria. I haven't tried that one. So in Brussels, we have Maastricht, Solito. There are some friends of yours. And Leffe, and then there was on Q&A, somebody was, Tina was drinking some for the drummer, which sounds really cool. So we have some American IPA. Then we have, let me see, yeah, some people from Amsterdam drinking Amstel, okay. Boston, we have people from Boston, that's very nice. Utrecht, a lot of people from Brussels, so that's, yeah, very nice, okay.
But somebody from Germany, from München.
Ah, somebody from München,
yep.
Nice. Munich.
Yep.
And then we have some very local patriots from Etterbeek. So nice. Okay, so let's move on. We have two awesome speakers left. So Milena, well, we let Milena join us again. We kind of need her for to continue. So I'm going to wait a little bit more. Yeah, let's see. So Gerardus Winterbier from somebody. And about the dog, I'm just letting Johan comment about the dog. What's the name of the dog?
Laika. Ha ha ha, no. I have no clue. I was just curious to what people would say.
Okay.
Of course, I don't know. It's not that I can talk with dogs, and especially not over internet. I tried to talk to Google, but it's.
You only tried over internet?
Yes, I'm afraid of dogs, actually.
Okay.
So I prefer virtualization.
Yeah. So I was expecting Joris to disappear, but it seems that Milena disappeared.
I'm here.
Okay, Milena. Great. Welcome back. So let me introduce Milena. Milena comes from the family of a psychologist, and she's a black sheep because she wanted to, just rebel against her family and do something totally else. So she went and studied engineering, and more specifically, she did medical engineering. She did biophysics, PhD in neuroscience, all in Belgrade, Serbia. And after that, she was in Finland, Austria, Italy, Spain, and now she's in Amsterdam. I know Milena for a really long time. We met in this science center we have in Serbia. It's a really awesome place. It's called Petnica Science Center, where you, as a high school student, can go and do a lot of science, and you can listen to a lot of super interesting speakers. And among really awesome speakers, Milena still stands out for me. I remember her coming, and she was originally supposed to give 2 hours talk, and but then she gave another one and another one, and I think we ended up with like 3 talks from her. And we were all talking that she has photographic memory because like nobody can just kind of remember all this interesting information she had in her mind. So nowadays she's leading a startup called Triega, and she's also still a researcher with Complutense in Madrid. She's a proof that you can't run away from your family heritage, because after all this engineering, she's using all her powers to actually study depression.
Yeah.
Her poll is here, so please fill in. Do you know somebody who suffers from depression? Did you have your things? So Charlotte, maybe we can see the results. Okay, so here are the results. And Milena, please, I'm leaving you with the results and you can start.
Okay, just to share my screen. Just a moment. Yeah. Can you see my screen?
No.
I shared that. Just a moment. Sorry. Oh, sorry. My computer is. Yeah. So I can share my screen. So yeah. Again, sorry.
Perfect.
Can you see that?
Yes.
No? Okay. So I have problems sharing. Yeah, okay. So I'm going to start with talking about depression.
Just as a remark, we don't see like the full screen. So we saw just part of the, so it's not full screen, that's just the, so maybe you could just like click on the arrow on the right to, yeah, that one, no. Just to have, you know, the menu, yeah, that here. Yes, that one. Thank you, Jose.
Okay, can you see it now?
No, can you see the annotation maybe?
No.
Just so just on the right, a little bit on the right, then go down.
Yeah.
Yeah, that one. No, just a little bit on the right.
The panel, you mean?
Yes, the panel. Just to
all in the middle of the screen.
Panel.
Left up.
Sorry, not sure.
Arrow.
Yeah, sorry. Share. Yeah. Can you see this?
I mean, we see actually the thing is that we see the presentation, but it's so it's in the PDF viewer.
Yes, it's PDF, unfortunately.
Yes, and no, that's not a problem. It's just that there is a panel, and because of that, we don't see the full presentation. So it's written like export PDF, Adobe export PDF, and all of that. And there is like something to
because I see something which is yeah.
You probably don't see the same screen. So maybe if you share again, then you can choose the panel.
Okay.
You can choose the window with the full screen.
Sorry, something is not working. I'm sharing my screen, but there is a problem with this. Sorry.
So maybe you can share the desktop. And then minimize the DataBeers. I mean the PDF.
Yes, I will do that. Just a moment. So,
okay, don't worry. Take your time.
Yeah. So, can you see this?
Nope.
Share this one. Can you?
No. You just start over. Just unshare and start over and try to choose the window.
New share. And then I have this. I'm really frustrated with this.
Okay, just relax. It's fine.
Okay. Can you see this now?
No, we are still seeing exactly the same thing.
Isn't it like F10 or F11 to make full screen? I don't remember, but
yeah, F11 maybe.
F11, can you?
No, I suspect there is a window with a full screen, and then there is this window, and you're sharing not a window with a full screen.
Or zoom out a bit, then it's smaller, but it.
Yeah, just zoom out.
At the top of your screen, there's a green button at the top of your screen. do the full screen of
new share. I start new share, and I do this.
No. At the top of your presentation of the PDF, of your window, there's the closing button, the red button. There's a yellow button and there's a green button. To close you click on the green button. No.
I just did completely different thing. Yeah, sorry. Sorry for waiting. This. Start sharing. Green. Okay. Can you see this?
It didn't change.
No, it's the same again. So can you just zoom out? Just put the minus, and then we can see.
So do you see my mouse? Like here, there's like this little thing. Can you click on this?
Sorry,
I don't know if my mouse is visible.
Approve. Yeah.
Yeah. Do you see where my mouse is moving?
No,
on the right.
Just a moment. I will try new share and do this. I'm not allowed to do this. Oh, sorry. Something is wrong. No. Can you see the slide depression?
Yes, but not the whole one. So if you just zoom out, we'll see the slide.
Just a moment.
If you just make it a little bit smaller, so go to like 80% or something, then we will be able to see it.
I'm trying, but it doesn't allow me to do this for some reason. Can we do this? Because
Or press the minus button. Does that zoom out?
Okay, I just say we move on. We see enough of the slides.
Yeah. So whenever I want to do minus, it doesn't allow me for some reason. So
maybe what we can do, Milena, is you can send me the presentation on DataBeers, the DataBeers mail, and I can just share my screen and try on my side.
Just a moment. I'm trying to do this. Stop share, and then I will do again. Depression share. Can you see this now?
It's the same.
Can we proceed with this? Because
yes.
I think it's going on.
Let's just go on. We see enough of this.
Okay, because we are wasting time. Sorry. So I saw the polls, and 80% of people know someone who is depressed. So if we observe the WH data, 1 in 4 persons is going to experience depression during her life. I'm saying her because to be a woman is 50% more a risk to suffer from depression. So the problem here is that how they do that in psychiatry. The problem is diagnostics. For example, if you think you're depressed, you go to someone who is, let's say, family doctor, you say, I'm depressed, and they give you antidepressants without any further notice. So the thing is that the problem is very old-fashioned classification. So if you show the symptoms, which are being really low, cannot sleep, having problems in functioning, escape from social contacts, etc., you already qualify for depression. But majority of people, due to stigma, don't even really report what kind of problems they have. So we realize that the nosology of psychiatry doesn't have so much in common with science, meaning that they rely still on report from a person who is suffering, and they rely only on that questionnaire and the impression of psychiatrists. So since I'm coming also from biophysics field, I realized that they are not using any objective biomarkers. So the thing is that there is a novel field called computational psychiatry. It is actually a marriage between computational neuroscience and psychiatry. So the basic idea is, can we use results of any kind of neuroimaging and data mining to find out the connections and some features which can really tell us whether a person is depressed or not? Because the whole time listening of previous panelists, I was trying to figure out what the connections are. For example, with dementia and with all the talk, whether the question we are trying to answer is significant enough. There are two key things that I want to state here. For example, number one causing death and disability is still cardiovascular disease. And number two is already depression. Because, for example, more than 10% of people in European Union are having problems with depression, reporting or not reporting. And when you find out the polling and, the field data, you find out that even if people report that they have problems with depression, only between 11 and 30% of them are actually having help, and they're getting better within the first year of the therapy. So in this slide, I just want to illustrate the history of computational psychiatry, because even when they start with neuroimaging, they found out that they have something that we call unwarranted optimism. Because we find out what technical problems are with application of machine learning in psychiatry in order to translate that to clinics. So we figured out that that branch of research, which is data-driven, is much more promising. And we turned to much cheapest way of detecting something from brain. So then we go to EEG, which is the oldest form of neuroimaging. They started in 1924. And this first slide is actually showing you if we use electrical signal from the brain and we chose feature from that signal, you can see that data are really separable, because the red dots are people who are healthy and the blue dots are people who have problem with depression. So let me tell you, I just actually merged something which is coming from complex systems dynamics, which is complexity studies, and data mining, meaning that in whole medicine, old-fashioned signal analysis coming from spectral and time series analysis are prevailing. And results coming from complexity studies are still reserved from research, for research. So if we use nonlinear analysis of electrophysiological data, then we can really solve that task in sufficient way to become useful. So my idea a couple of years ago was to try and show that if we characterize that signal with some of nonlinear measures, I mean, fractal measures, entropy measures, whatever nonlinear measures, and we apply any of popular classification models, we can have really high accuracy, which you can see on this slide, that if we apply multi-layer perception, logistic regression, support vector machine with or without, with polynomial kernel, then decision tree, random forest, naive Bayes, every classifier actually returns pretty high accuracy. The only condition is to characterize that signal with some nonlinear measures. I will try to explain this. If you focus on any electrical signal from the body, let's say electroencephalogram, electrocardiogram, electromyogram, when you go and analyze that, you realize that those signals are nonlinear, non-stationary, and noisy. And at one point you realize if you're applying 19th century methodology, which is reductionistic, like Fourier, you're actually damaging the signal and you cannot get real features out of it. But if you apply at least fractal and entropy measures, you can characterize signal that well that any classification can do. So we use only principal component analysis to choose what features we are going to introduce to the classifier. And everything we did is actually pretty well. It's everything above 92% accuracy. Another false notion among researchers is that they can choose, for example, only 1 or 2 or 3 or only frontal electrodes. And the fact is that in depression, you can see that on all cortex. I will go just a step back to explain to you why that works. For example, when you observe a brain of person who has depression, they have the problem with deep white matter connections between prefrontal cortices and limbic ones, which shows to be problem with functional connectivity. So everyone who has depression has a problem with control of affect, control of emotions. So they cannot, they are stuck with focusing on negative feelings and negative emotions. And as a consequence, you can see the change of the level of complexity of the signal of whole cortex. And in this slide, we actually show that every single electrode on the whole cap is contributing to the detection. So on the next slide, actually, we tried to show to the people that there is already enough research showing that detection is possible. And we analyze raw EEG signal because whenever you do any kind of artifact removal, you're introducing another artifact. If you're filtering, you're actually getting rid of important information from the data. Then when you analyze those epochs of EEG signals, the best recipe is actually to look for the sections of the signal which is not noisy and analyze that. And after that, you perform time series analysis and feature extraction. And on the end, the classification task is to see whether the person is really depressed or not, because their intrinsic dynamics is really different. Why we can say that? Because if the deep connections in the brain are disrupted, the signal is finding the way how to solve the same problem another way. So the dynamic of the brain of a person who is depressed is in dynamical space really different, and you can detect that. So this is some kind of review, because there is already enough publications to show that it is possible to detect people who are depressed. Why am I showing this? Because it is already enough data that we can do that clinically, and people are still not applying it because there is a gap in perception. Because when you ask someone who is medical practitioner or a doctor, neurologist, psychiatrist, they simply have a problem of understanding the notion of complexity and nonlinear analysis, because spectral analysis is deeply rooted in every single piece of equipment they have, and nonlinear analysis is still not there. It is still reserved for research. So what I wanted to discuss here is problems which are actually preventing us from applying that broadly. So like in the first presentation that we listened tonight, very modest sample sizes is the problem number one, because this is the reason why we left the idea of MRI use for depression detection, because the number of features is usually much larger than the sample. For example, you have only 30 persons scanned or maybe 50, but you never see the sample which is larger than 100. So the only solution for that is to collect more data and to start collaborations between different institutions who are collecting that data. Then there is another problem that we are facing every day. This is GDPR data collection. Because, for example, in Netherlands, even if you want to see your own data, they're not going to give you anything on paper because they are protecting your data from yourself. And every institution which is trying to anonymize the data, they are still afraid because there is the way to find out about the identity of the person under the study. And we opted to focus on electrophysiological data because according to GDPR regulations, it is not possible to reconstruct the identity based only on electrical signal from the body. And there are several Dutch initiatives which are trying to open their databases to other researchers so they can standardize their methods and develop further. And one big problem with doing research in this field is the lack of proper validation. So my colleagues and I are actually advocating specific mandatory conditions even in publication. Because when you read the literature, which is publishable, I mean, the things which are already published, you cannot simply repeat the experiment because first, there is not enough data about how precisely they did the research. And second, you know, the problem is that small samples are so ever present in every research, then people have to realize the generalization is in close connection with the small samples. For example, if you do the training phase, you realize that your algorithm is trying to recognize exactly the first sample in every other sample. And we call that nuisance data. For example, you do the training phase, and after that, when you show to the algorithm the test data, they're trying to recognize intrinsic connections and even a noise in another set. So this is something we must prevent in order that all these concepts would be translatable to clinical research, to clinical application, actually. So one thing that is solving unwarranted optimism, we call unwarranted optimism the results which are above 96 or 97%. Because if researchers who publish those results did properly their validation, they would probably have much less accuracy. I think now, according to what I know and the data sets that I saw in the last 5 years, that even 80% or 85% is accuracy high enough to solve that task. So personalized medicine is exactly what fits with this model. You're actually seeing the patient who is saying that this is depressed. And before giving him or her antidepressants, you should do the classification with the electrophysiological data to see whether that person really needs medication which has really dangerous side effects. So I just made this slide to show what overfitting is, because overfitting happens when you do this task with a really small sample, which is not enough for giving you opportunity to translate that to clinical research. You can solve this only with collecting more data and doing certain steps in analysis to prevent falsely high accuracy. So I didn't have more slides, but what I can tell you is that regarding to the second talk that I heard tonight is practical application in business. If you start talking about complexity analysis and machine learning at the same time, they're lost. Business people simply cannot comprehend what is complexity. And I can say that we as the researchers are still don't have a consensus what complexity in electrophysiological data are. And the thing with all this project with detecting depression, I can see the only solution in collaborative effort with many different universities gathering the forces to collect more data and train their samples so they can really show people from clinics that this is really feasible. For example, I know some numbers from Holland, from Netherlands. For example, treating a person who has depression within the first year costs insurance something which is more than 80,000 euros. And this is only talk therapy. So introducing some really objective biomarkers for depression, which are already there, because there is already sufficient research showing that this is possible, it has to happen really soon, because the present system is really unsustainable. It is wasting too much time and too much money, because the waiting list for someone who is depressed to see the therapist who can help him is between 6 and 9 months. So I'm, thinking whether you have any other idea about alternative therapies, because they are also there. But this composition of complexity measures and machine learning is really yielding a solution for mental health detection, which can be much better than it is now.
Thank you, Milena. I think this is a very good point to stop. Do we have questions from Milena, please?
I saw a question in the chat from Joris, so maybe he can ask her.
Yeah, I had a question. So I was wondering how feasible it is to use these techniques to start detecting depression on a large scale. Is it expensive to do? Is there enough equipment to do this? How would this work?
Yeah, well, very logical question. Actually, I wanted to tell that during my presentation that we left very expensive MRI, because if you want to check the phases of development in somebody's treatment, you cannot send the patient every two weeks to MRI, because it's simply more than 1,000 euros. So that's why we shifted to much cheaper methods of detection, which is EEG, because every modern hospital has this EEG. And even cheaper than that is portable ECG device, which can record a heart rhythm. And it is also possible to detect depression from ECG, which is cardiac signal. So we are basically working on that at the moment, because we are aware that many portable devices are on the market. And with advent of many devices, it will be feasible to say that outpatient monitoring is really at the reach of the hand. Because I believe that even this crisis with COVID-19 shows us that telehealth is really something we need badly, because the regular care and diagnostics are simply non-sustainable.
Thank you. More questions from the audience or from the other panelists? Nope, I don't see anything.
So there is somebody who asked a question in the Q&A.
Okay. Can you let them speak?
Hello, Milena. Thank you for the presentation. I had a question from a point of view of a machine learning researcher. I see that the approach you've taken and you presented is to classify depression like as a binary problem. Either the subject is depressed or not depressed. But as far as I know about depression, it's something which can be represented more like on a spectrum, or with different degrees, different level, and I was wondering, is there a particular reason to treat it as a binary classification problem, or just to simplify the problem, or are there any other motivation behind that?
Yes, good point. The central question in treating depression is whether that depression is melancholic or not melancholic. Because people who have recurrent or melancholic depression, they have somatic features, like they cannot sleep, they cannot eat, they cannot function. These are all unwilling things controlled by autonomous nervous system. People who have one-off depression have much slighter symptoms, and it can be treated with SSRI drugs. So for me, from the point of usefulness of the whole methodology is to detect whether people really have those features going in that club, which is really hard recurrent depression. This is what we are detecting. So I was thinking about the whole methodology as the decision support solution, because if you're a therapist and you have a patient, you need to know very early whether that person is really recurrent depression or something light, which can be treated with one medication. So as you noticed, it is a binary problem because the treatments between the two are really completely different. For example, people who have recurrent depression, they react well on electroconvulsive therapy and tricyclic depressions. And people who have one-off depression even recover without the treatment. So the real thing is that we can classify that, which would be really beneficial if we detect early. So I hope I answered your question.
Yes, indeed. Thanks. It makes more sense now why there is this clear distinction between the two classes. And so I would agree in tackling the problem as a binary classification problem. Thank you.
Yeah, thank you.
Okay, thank you, Milena. So, so let's move on to our last speaker for tonight. So Sam, let me present Sam. Sam, so I'm really glad we already talked about complex systems until now, because this is exactly the point of Sam's talk. Sam is one of the leading scientists in network science. If you look at his Google Scholar page, you'll see that he published in all the important journals like Nature, Science, PNAS, Science Advances, Lancet, all of them. He got 1,400 citations only this year. So he is the right person to ask things about the pandemic going on. And he's been doing it before it was fancy, okay? So he's been doing this for years. And so there's been a lot of information and misinformation going on. And what Sam is going to focus here is how networks are contributing to this pandemic, or why it's important to see this pandemic as a complex system. So please, we have only one question for Sam. Let's see the poll results. Oh, okay. It seems that a lot of people do know what's complex networks. Everybody had graph theory at school, so that's a good start. So Sam, I'm letting you take over.
Great. Well, thank you very much, Milena, for the very overly kind introduction, and the rest of the organizers for inviting me, the speakers for giving really 3 very exciting and interesting talks. I really enjoyed them and learned quite a bit. So it's been a lot of fun. And to everybody who's joined us, my only wish is that we were together drinking beer. But it is really a pleasure to be able to do these kinds of things, even if we're far apart. It kind of reminds me that there's a whole world out here, and there are things to be excited about and connections that we can still build with each other. Even when we're remote. So thank you all for joining us for this. All right, let me share my screen here. And everyone can see my screen?
Yes.
Yes.
Great. Okay. So one of the things that our group does at Northeastern is to build mathematical and computer simulation models of infectious disease outbreaks, and then try to translate the results of those models into what we hope are improved public health and clinical decision-making practices. And for us, one of the things that seems to be very clear about COVID-19 is that part of the answer to why it became a pandemic is because it is very much a complex system, and it is operating as a complex system such that if we want to control the pandemic and we want to do so in such a way that minimally disrupts our economy, minimally disrupts our social lives, has minimal effects on our mental, physical well-being, on access to education and social services, we have to understand how it's operating in the context of this diverse socio-technical system that we've built across the globe, and how a pathogen like COVID-19 is able to spread so efficiently in some parts of our society and so inefficiently in other parts of our society. Why some countries are able to completely prevent COVID-19 from taking hold, others are able to bring it back under control, and yet others like the United States are essentially just allowing it to run rampant, causing untold trauma, huge rates of morbidity and mortality, and massive economic consequences. And I think that really, unlike many other pandemics, which are also complex systems, COVID is very unique in how acutely it feels the variability in our societies, how acutely it affects and interacts with our behavior, and how it feeds back on the ways in which we communicate and interact with each other. So just very briefly, and I think for many of you, this is review. So from my perspective, what do I think of as a complex system? Well, I think of a complex system as something where there are many parts, often they're distributed, and critically, that the way in which they interact may lead to emergent properties like a pandemic that could be relatively difficult to predict by just looking at some of the individual components themselves. Or that if we have the pandemic that occurs, taking the pandemic and trying to dissect from the top down without also incorporating information from the bottom up and maybe meeting somewhere in the middle, it's going to be very difficult to disentangle mechanism from noise, or mechanism from all the other ways in which we could have had a similar pandemic trajectory play out. And indeed, some of the things that we've heard in a previous talk suggest that this kind of feature, right, is quite common across many of the things that we're interested in society, as a society and as scientists and in the business world, is understanding how we really take parts and dissect them and then build them back up into the whole, how we take the whole, dissect it down into the parts, but I think actually really critically, when we can and cannot do that. So when we are faced with something where we really are not going to be able to dissect from the top down, and instead we need to try and understand how these individual components interact. And so here I'm just showing you some examples. John Snow's 1855 study of a cholera outbreak in London, I use as an example of the effect of socioeconomic inequality on disease burden, which is one of the biggest features of infectious diseases. You look in a country like the United States, disproportionately affecting communities of color, indigenous populations, and it's as a direct cause of racism, xenophobia in the country that we're seeing these kinds of things. The effects of the evolution of the pathogen, the effects of human behavior, the effects of human mobility and social networks, and how all of these things interact in different ways to result in countries like the United States experiencing COVID-19 as one of the worst causes of mortality and economic despair in generations, and other countries like Vietnam, Thailand, Mongolia, Senegal, Nigeria, Japan, Singapore, China, largely controlling the outbreak with non-pharmaceutical interventions. And so then maybe just to be a little bit more specific, and I think these are really critical features of COVID, and if you're interested, there's a fantastic piece in the Atlantic called The Overlooked Variable. This overlooked variable is the key to the pandemic, about this sort of effects of super spreading and how it challenges and breaks, especially our Western public health models around how we should respond to pandemics that are largely based on influenza. So transmission of infectious diseases like COVID are highly heterogeneous. Few people infect many, and many people infect very few. They're dependent on spatial structure and the underlying population. Again, especially true for COVID-19. You have stark differences in the population connectivity in urban areas like Sao Paulo that differ from more rural but still large cities like Manaus, and it explains quite a bit of the variability between places that experience sharp, intense outbreaks and other places that experience, although overall a larger proportion of the population are infected, long, broad, persistent epidemics. They're unequal in terms of who's most affected and why: age, race, income, especially true for COVID. And of course, we're biased because this is how we think about the whole world. This is how we think about biology, the universe, is that they're best conceptualized and understood as a network phenomenon where we need to understand how parts interact and come up with these emergent properties that we call pandemics. And this slide I actually took from one of the researchers in our group, is the individual in the lower right here named Brennan Klein. Brennan is one of the rising stars, in my opinion, in complex systems, data science, network science. And so be on the lookout for Brennan. If you ever see his name, drop everything and rush to hear what he's working on. And ultimately for me, what I want to understand is this map, right? So this is the heat map of COVID-19 cases by country. And there are places like the United States, much of Europe, Western Europe at least, South America, that have been hit quite hard. There are other places, China, Japan, Vietnam, Thailand, Nigeria, Senegal, Uruguay, that have not been hit hard. Places like Australia that have repeatedly controlled introductions, places like New Zealand that it's largely business as usual. And this is largely because of how the governments have responded, differences in human behavior, differences in the underlying effects of racism, xenophobia, classism, leadership or lack thereof, belief in science, accessibility of data, data-driven decision-making. These are the things that are driving much of this variability. And if we're going to return to any sense of normalcy in the near future, it's going to require us to embrace these challenges and understand how this map came to be so that we can intervene in such a way that we are able to bring the outbreak under control with minimal disruption to our mental, physical, social well-being, to our economies, and to our health. And a lot of this actually starts with SARS. And SARS is very important in the network science and complex systems community, in part because of what didn't happen. So this is a quote from a nurse in Canada: "If we don't have a vaccine, yes, we are all going to get it. We are all going to get it." And of course, that didn't happen. And it largely didn't happen because of super spreading events. So SARS has an average number of secondary infections that's large enough that we would predict it could cause a pandemic. But it turns out that this average number of secondary infections for SARS is largely driven by giant super spreading events. And that if you can intervene to prevent the super spreading events, the epidemic will rapidly die out. And the establishment probability, the probability that somebody walks off of an airplane infectious with SARS and sparks an epidemic that sweeps across the United States, is highly stochastic. Many of these introductions will lead to 0, 1, 2, 3 other cases, even with minimal to no public health interventions. And you really just have to prevent the super spreading events or not get unlucky. And this tells us quite a bit about many infectious diseases. So here's a picture from 1918 flu. Here's a picture from the 2014 Ebola outbreak in West Africa. Both of these pathogens have approximately the same number of secondary infections too. 1918 flu infected 500 million people. Ebola in West Africa devastated Sierra Leone, Guinea, Liberia, had huge economic consequences, although it only infected 30,000 individuals. And the difference between these two pathogens, in many cases, boils down to the fact that influenza marches along pretty regularly. Everybody infects 2 other people. Whereas Ebola, pretty much everybody infects 0 people, and then occasionally somebody infects 20, 30, 40, 50 other people. A paper by Jamie Lloyd-Smith et al. Helps us conceptualize some of these pathogens. And there's been quite a bit of very influential work in the network science community. Some of you on this call have certainly been a part of that. And the way that Lloyd-Smith et al. Thinks about this is, imagine on the x-axis, I could rank the infectious cases today in Boston. based on how many new cases they're going to give rise to. And that's the y-axis. So this 1-to-1 line is a homogeneous population if we were just randomly mixing with each other. 50% of the people infected today are going to infect 50% of the people that are infected tomorrow. As these lines bow up and to the left, an increasingly small percentage of the people infected today infect an increasingly large percentage of the people infected tomorrow. Pneumonic plague, monkeypox, smallpox, measles, SARS. And if I label this with Ebola and 1918 flu, I'm rounding a bit here, but Ebola is very much like SARS, highly dependent on super spreading events. And without them, it rapidly goes locally extinct, whereas influenza marches along very, very regularly until you deploy a vaccine. COVID is in this very problematic space where it is pretty reliant on super spreading events for sustained transmission, but not entirely. It's a little bit more like influenza than it is like SARS-1 and Ebola. And it's just enough that you can control it with active testing, tracing, isolation, and quarantine. But you can rapidly let things get out of control if just a few too many transmission chains take hold in a country. This mixture of a reliance on super spreading events and deterministic transmission is also part of the reason it breaks so many of our Western public health interventions that are either based on influenza or Ebola and SARS, where we can stop them with temperature screening. Individuals are not infectious until they have symptoms, and the symptoms are typically debilitating, where COVID is right in the middle. We can use complex systems. We can do backwards contact tracing and cluster busting like they do in Japan. We can do highly active case investigation, testing, isolation, quarantine, like they're doing in Australia, like they're doing in New Zealand. But once it takes hold, it's very, very difficult to control without lots of buy-in and coordination across the public health response. And so the other feature of this is that because of its effect of super spreading, because of the effect of social network variation, it acutely feels our variability in contact structure. And so one of the studies that we published is looking at China and at Italy, two countries at the time that had experienced complete waves of COVID-19, how local variability in crowding affects the sharpness of the epidemic curve, such that areas that are less crowded and comparatively more rural have sharper and more intense epidemic curves, and areas that are more urban have longer, broader epidemic curves. So think Manaus versus Sao Paulo. Think about a rural area in the north of Italy versus London or Milan. This variability between places like Madrid and places like Ulaanbaatar in Mongolia. And we can think about this in the context of a simulation where we instantiate different hierarchical populations that people live in, connections of households that are connected into neighborhoods that are connected into cities, or one very tight-knit population living in a more rural area. And we can simulate COVID-19 in these different communities and see that in the tight-knit rural communities, you get this relatively symmetric, sharp epidemic curve that looks like, if you're familiar with the susceptible infectious recovered SIR models, the classic SIR model in a well-mixed population. Whereas in the right, what's really happening is you have lots of little waves happening in different neighborhoods that because of stochasticity and reporting bias, you get this long smear, this long, broad epidemic. And we can project these estimates in different populations throughout the globe using high resolution mobility, population, human behavior data, and look at how the substrate of these human contact networks is going to interact with behavior responses, interact with the coordinated or lack of coordinated response at the government levels, interact with the availability of testing and personal protective equipment to drive variability in the establishment probability of COVID-19 and our ability to control it once it becomes established. Lastly, I'll just mention this because this is a conversation about data. One of the things that we're trying to do is address the critical data gaps that are associated with COVID-19. We've bootstrapped an international consortium that is curating an open individual level anonymized data set that now has over 3 million deduplicated, geographically resolved cases from 160 countries of COVID-19 going all the way back to January, so that we can further understand how differences in age structure, differences in the effects of racism, differences in the way that governments roll out interventions has played out in terms of the epidemic in different countries. And this is work that's in collaboration. That is the wrong slide. This is work that was highlighted by the New York Times that said an informal network of hundreds of volunteers has assembled records for more than 1 million cases in 142 countries, representing, this is back in June, the single most accurate portrait of the virus's spread in existence. I want to end by just reminding everyone, and this is data from the US, that COVID-19 is hitting Black and poor communities the hardest, underscoring fault lines in access and care for those on the margins. This is a very powerful piece in the conversation by Grace Noppert, showing that poor communities are hotspots for COVID-19. For example, the Washington Post reports that 14% of the population in the state of Michigan is Black, but 40% of the COVID-19 deaths are among Blacks. The population adjusted rates of deaths in the United States for indigenous populations, Hispanic, Black, and early generation immigrants are 2 times what they should be given the population percentages. And this is what leads us to conclude that COVID-19 became a pandemic because the world doesn't understand complex systems, doesn't understand complex networks. In particular, maybe the Western world doesn't understand complex systems or networks. Maybe it's just the United States. And that because so much of our public health response is rooted in a misunderstanding of the way diseases work, way complex systems work, and rooted in a lack of willingness to address centuries now of racism and xenophobia, we are in this position where a country like the United States that ranked first in terms of pandemic preparedness in 2019 has now experienced over 20% of all the cases, over 20% of all the deaths. So again, I want to thank you all for your attention and for joining us. Thank the other speakers and the organizers, and happy to take questions at their time.
Thank you, Sam. So we are now going to move to the questions for Sam. Anything in the chat? Nope. Panelists?
I have just a question personally. Yep. So I was wondering, one of the things that we saw in your presentation is that there is a huge how to say, influence on the urbanization and these kind of things. So is there any kind of latent variable that could also explain, like such as also census or the connection to, for example, we saw especially in Africa and these kind of things, and also in China, there is a lot of villages that are not necessarily connected to the administration and this kind of thing. So could that also explain this variation in the also explaining that we don't necessarily see who has the virus and these kind of things?
It's a wonderful question. Thank you very much. So just to quickly restate it. So urban-rural divides, how much of that has to do with differences in the actual social contact networks, for example, in urban versus rural areas versus how likely COVID is to show up or biases in data systems. So I think what's really important to separate out, and I did make this clear, is that our analysis is about what happens after it shows up and in places where we think have pretty good surveillance systems, or at least they're equally biased in the urban and rural areas. And so after the disease shows up and takes hold, you get these sharp, intense outbreaks in places that are more rural. It's really actually places that have a particular kind of social network structure that just kind of tend to be more rural. So I'm using that as a bit of a hand-waving example. But what's really important, right, is that rural areas are less likely to have the disease show up. And in fact, it's one of the things we see in the US, we see in many other countries, is that the rural areas get hit later. And oftentimes they get hit later because of complacency. They equate their safety with something else aside from the fact that they're just lucky in the sense that it's going to take longer for the disease to show up. But maybe once it does show up, it can be a lot more intense. We do also see, and this is actually much stronger in indigenous populations, huge data biases. So there's essentially no data coming out of many of the indigenous communities across the globe that we know are being hit very, very hard. Mexico is really conspicuous. If you look at the state of Oaxaca, there are big blank spots in the mortality. And it's not because people are dying, it's because these are heavily, a high percentage indigenous populations in Oaxaca that are just not reporting because the state's not monitoring, right? And so we know one of the ways that states disempower communities is by not monitoring the health burdens there. And so we're seeing that kind of play out on a global scale. But yeah, I think all of those issues you said certainly are interacting. And I was massively oversimplifying in that kind of whirlwind tour through what we were doing.
Thank you.
We have a question from the audience. Vasivalod, do you want to ask it yourself?
Yeah,
Can I ask
hi,
something?
Sam. So basically the question is whether you have some nice ideas, obviously not extensively, but how to figure out where we have some misinformation on, for example, the reported number of cases. So to have an idea who is kind of cheating while proposing the stats which are far from the reality.
Yeah, great question. So the question is about kind of misinformation around cases. I think it's most approachable for me in two ways. One is with mortality data, because they tend to be, not that people aren't biased in terms of how deaths get reported, but countries tend to be much more data complete around all the deaths that happen. And so you could imagine, and maybe we can't do this until it's retrospective, but building models that actually try to predict who really died of COVID, given a nearly complete census of deaths that happens in countries. So I think that's something that we're going to be seeing a lot of as we move forward from this. I think the second is by blending across different data sets, right, that we think are all giving us different kinds of information, sometimes complementary, sometimes not. A really powerful option for this is leveraging genomic data from COVID-19 pathogens and trying to reconstruct the epidemiological dynamics using the mutational differences across the pathogens and comparing that to epidemiological data. But I think, you know, the sorts of machine learning approaches that many people here work on, where you're really trying to blend across data sets, is going to be another really powerful area. But I think it's a huge problem, right? I mean, we're really trying to ask this question: how do we decide whether the data are just missing, right? And that's a really hard question to answer, especially in the context of a lot of our statistical methods, where in some ways, if we formulated it that way, we'd be trying to prove a null hypothesis. So I think in many ways it forces us to adopt different ways, and many of you are doing this anyways, but like adopt different ways of thinking about how we approach questions and how we approach, you know, sort of scientific analysis of data.
Yeah, nice, thank you.
Can I ask something?
Milena, you had a question?
Yes, I was thinking about Spain, which has a really high percentage of very old people. So this varies across the countries. Did you normalize for that? Because, you know, older people usually has a higher risk of, you know, coincidence with some other illnesses.
That's a very good question. Did we normalize for age? We do to a certain extent, but not, I think, exactly in the way that I would, and I know individuals are working on this study, but one of the things that consistently shows up is that the prevalence of cohabitation between grandparents and younger generations is a really strong predictor of mortality variability. And that seems to have been something in Spain and in Italy and other places in parts of the US where you would experience very, very high mortality. I don't know that there's been a large scale comparative study that's really kind of integrating out all of the other reasons that we might expect to see that. But I think that's part of the answer. I mean, it's one of the things that people don't talk about enough with Sweden, right? Is that setting aside whether what they're doing is working or not working, let's just set that aside. You know, their average household size is really low, like it's like almost less than 1. And so if what's working in Sweden is just having low average household size because a third of the cases happen in the household setting, that's not really going to be portable to other places, right? And so I think one of the things that we really have to try to answer is like, is something working because it's a good policy, or is it working because of the kinds of society that's present that's just not really going to be replicable in other places? And again, that's why I think this kind of complex systems, complex network lens is really the only way we're going to get an answer to those questions.
Thank you.
Thank you. Any more questions? If not, I would have one. It's yeah. You said that at least from US, the Black and Hispanic are hit much harder than whites, but also Black and Hispanic have much lower socioeconomic status, so are more likely to work in you know, as a cash register, or the things which are you know, just kind of more in contact with other people. Did you control for socioeconomic status, and does this prevail still?
It's a very good question. So there's certainly a direct effect of socioeconomic status on disease burden that cuts across race, ethnicity, and other factors. There is also an interaction between socioeconomic status and race. And this is not analysis that we've been a part of, but there are many papers showing a direct causal relationship that links the effects of racism to socioeconomic status in these communities. And then you have this kind of indirect effect modulated by socioeconomic insecurity and access to healthcare on disease burden, on COVID-19 and those sorts of things. So the short answer is it's very complicated, but the strength of the epidemiological evidence that racism and xenophobia is the underlying causal feature of this is very, very strong for almost anything anyone has ever looked. And for COVID, we see the very similar pattern where there is also a general effect of socioeconomic inequality that cuts across race and ethnicity, but there's also a second effect of that as well.
Don't get me wrong. I believe that racism is a really big problem in the world. I'm just wondering, is this the cause through socioeconomic status? I didn't even think about hospitals and their access to healthcare. I was just thinking about kind of jobs they usually do, and which is kind of more likely that they work like essential jobs than this. So I was just wondering if this is more explained by their socioeconomic status than actual pure, just racism. Like would it be consequence of racism before they get to the racism which put them in a socioeconomic status they are? Okay.
From my perspective, yeah. Well, I don't know that anyone has actually done the kind of detailed analysis that you're suggesting that would be required to pull those apart. I will say that I have such a strong prior on the organizational effects of racism in the United States that it would take nearly an infinite amount of data to convince me otherwise at this point. But whether it's like how strongly it's modulated by the effects of racism on socioeconomic status versus any direct effect of racism, I think is a very good question and exactly the kind of thing that we're going to have to understand. And it's going to vary by country, right? I mean, it's one of the things that so many of us are dealing with as increasingly international organizations, international groups, international communities, is like how we would define racism and what individuals are affected by that in the US is not going to be the same in other countries. And so how we come up with some kind of globally representative model of what we even mean by racism is really going to be very challenging and something that I think we're going to have to come together around as a global community.
I have another question. Sorry.
Yep.
It strikes to my mind something that I heard from a German researcher. They actually find out that the asymptomatic people were actually cause of 40% of infections. And they don't even know that they are infected, after a week. After that, they develop symptoms. What do you think would be the best way to model that? Because this is obviously something which is below the radar. And this is really a big problem which has to be tackled because asymptomatic people seem to be one of the biggest problems to predict anything. How do you think?
Yeah. Oh, sorry, but it's a great question. So just very briefly, right, so the questions about asymptomatic individuals. There's a couple of big challenges. So the first is that most of our contact tracing and case investigation systems are completely broken. So for example, in Massachusetts, we're only able to positively identify the source of 50% of the infections. And I don't even really necessarily believe the rest of the 50% either. And so part of the problem is like, how are we really ascertaining whether it's the asymptomatic infections or not, and how general is that? I think it's probably pretty general, but it's a big data. It's not a big data problem. It's a big data problem. The other is that there's two kinds of asymptomatic transmission. The first is individuals who are not yet symptomatic and infectious that infect people, but will become symptomatic. Mm-hmm. And that certainly looks to be about 40 to 50% of infections. The other are people who will never have symptoms. And they are certainly infecting other people, but what percentage of infections they're responsible for, I think, is a big question mark. Some of the viral count data coming actually from the National Basketball Association in the US, where they're following people longitudinally, suggests that the asymptomatic individuals do have a different viral dynamics and they clear it faster than the symptomatic people. So it may actually be the case that the truly asymptomatic people are not infecting as many as we think, and that a lot of it are people that go on to develop symptoms or have minimal symptoms. But that's one of the things that's really going to be very difficult for us to deal with. I do also think we have a lot of evidence building to suggest that you have to test people who are asymptomatic. It's what the colleges and universities in the US that are working are doing. But that also raises really challenging ethical questions around testing people, especially if it's invasive, that don't have disease. It's my favorite question I've been asked so far is, what do I think about the ethics of high resolution digital contact tracing? And my answer is like, I wish we could talk about that because nobody's actually rolled that out anywhere outside of like South Korea. And so like, I thought we were going to be having these like deep intellectual conversations about the ethics of high resolution digital tracing, but we're just not doing it. And so like we built all these systems and they never got deployed or they never got adopted. And I think it's because, I mean, back to the second talk, right? It's like people have to want to use it. And if they aren't going to use it and the governments aren't going to enforce it, then we're never even, we wasted all this, we didn't waste it, but like it's purely academic, the conversation around the ethics of it. And it's ultimately like a UX experience issue.
Okay, so one last very short question. If you have one thing you can tell to policymakers in any country, US or any Belgium, what would be just one thing?
We need a plan now for how we're going to deploy vaccines that are not going to be sufficient to generate herd immunity on their own, but through a combination of non-pharmaceutical interventions, contact tracing, testing, isolation, we can return to a new normal much faster by leveraging a low efficacy vaccine that doesn't have enough coverage. But if we don't plan now, we're going to be in the same situation that we're in now, which is like many countries just like not having plans for things and then being surprised when their lack of plan leads to disaster. So it's gonna happen. It's gonna probably be April or May when lots of countries start to have some access to vaccines, and that means we've got to be figuring this out right now. Although in the US, we've got to get over something that's happening next week before we can figure out if there's a future. So,
okay. Well, thank you, Sam. So this is the end of officially the longest DataBeers in the history of DataBeers, even when we have the live ones. So we need to know the winners of the beers. So Jose.
Okay, I didn't know it was going to be done immediately. We actually are we including panelists for the 5 beers being sent or not? Yelena, I don't hear you. I still don't hear you.
Yelena, your mic is on mute.
That's such a pity. I just had something. I said something very smart. So yeah. Okay. No. Yeah. We can include panelists. I guess panelists deserve a beer. But unfortunately, Sam, US is too far. But we promise you, when you get closer, you get lots of beers.
Okay. Then let me do the final count. This for sure. Also for sure. Only Belgium also. So then.
And close enough. Yeah, I think we can send beers to Maastricht.
Okay. Then, okay, let me see. We've got Jacopo. We've got Veselov, like help. Veselov, thank you.
Yeah.
We've got Anton Veenstra. And we have Joris. And who do I choose here?
Okay, we can send 6.
Milan, and but then Milenka and Charlotte.
So Milena and?
Oh, Milena, sorry, and Charlotte.
Charlotte.
Yeah. For the
no, I don't think we can send to organizers. So
yeah, but that was a panelist bit. Okay, then not Charlotte. Sorry, Charlotte.
Well, Lito is officially ex-organizer, so we can send it to Lito. I think.
Lito. Okay.
Okay.
Oh, you decide on that one.
Out of these people, Anton.
Yeah.
So if we don't find you in registrations and everything, please get in touch with us so we can get your details to send you the beers.
Yeah.
I think most of other people's we know how to contact. So thank you very much to everybody. I hope you enjoyed, especially thanks to the speakers. and everybody who survived this late for all these interesting talks. So then